Hello, biology people. Welcome to Miss Schimmel's class. Welcome to the genetics unit. Today we're going to talk about Punnett squares again, and I want you to remember that it's a tool that can help you to look at the possibility and the likelihood, the probability of some type of genotype, the expressed type of genes, and the phenotype, the physical appearance of that organism. What's new? What's different? This time we're going to look at incomplete dominance. Before we looked at complete Mendelian genetics, today we're going to look at incomplete dominance where you end up with something in between. I hope you remember from classical Mendelian genetics, if you have something dominant, you also have something recessive. Anytime you take two hybrids, you're going to end up with something being purely dominant, for example, being able to roll your tongue. You will have two that are also dominant, but they're hybrid for the trait being capital R, small r, and of course you end up with one down here being recessive, that cannot roll their tongue. This is a classic Mendelian genetics using a Punnett square. This is anytime you get two hybrid parents. Remember those fancy ratios of 1 to 2 to 1 and a 3 to 1 for dominant to recessive? This is going to be different for incomplete dominance today. Since we're now talking about incomplete dominance, both alleles code for an expression of the trait. The phenotype will appear as if it's blended. For example, if you took red and white, neither of the children would end up being red or white. In this case, we would appear pink. That's because there appears to be a blending. So if you think about colors, red is important. Blue is also important. We would capitalize the letters since they're important and strong to symbolize them. Remember that you need two alleles for each genotype. So in the case of blue, we would make you capital B, capital B. How would we write red? Remember that red is also important, so we would make you capital R for red. In this case, we have four capital letters. Red is dominant, blue is dominant, but if you mix them together, you're going to make red and blue make purple. And you're not going to have a purple gene, no purple allele, there is no P. Instead, we're going to end up being RB or BR, purple. Since red is not truly dominant over blue, we're still going to use capital letters to signify that importance. Blue is not completely dominant over red, so we're going to use B's to represent blue. Big B makes you homozygous blue. Remember that if you get some blue passed down and some red, you're going to be purple. If you get some blue and some red, you're going to get purple. There is no purple allele, however. Purple is a result of both blue and red. So I'm going to try to draw these in. Purple is a result of both blue and red. Here's the red, just added on the screen so you can see the genotype for a parent if they are red. There is no purple, no P's. That does not exist. Purple is a result of both red and blue. Purple is a result of both blue and red at the same time. It doesn't matter what letter you put first, one is not dominant over the other. Just saying it again for incomplete dominance. If you get some red and some blue, you're going to end up looking, phenotype, appearing purple in color. If you mix some blue and some red, you're going to end up being purple in color. There is no purple allele, no capital P. No capital P for purple. This does not exist. However, purple is a result of both red and blue at the same time. Your appearance would be purple in color. Purple is a result of blue and red together. Purple is a mixture, a combination. It appears blended because neither gene, neither allele, neither chromosome has complete dominance over the other. For an incomplete dominance Punnett square, the instructions say to cross a red with a blue. To be red, you must be homozygous. To be blue, you must also be homozygous. We're going to place the red parent here and transfer the alleles down as if this did meiosis, as if this parent created four separate genotypes to pass down to their offspring. Actually, just the four alleles to make four separate gametes. Say, for example, this is the mother, and she passes down her four eggs to her offspring. Going back to the instructions, it said cross a red with a blue. Remember, blue must also be homozygous. We're going to make this the father over here. Whenever he goes through spermatogenesis, oogenesis, whenever he does meiosis, remember that he would make 4 separate sperm cells, and he can only pass on half of his information to his babies. In this case, he's blue, so he passes down the blue allele. Whenever these children are created from the fusion of the egg and sperm from the male symbol and the female symbol, all 4 of these offspring are going to be ending up as purple in color. Remember that the genotypic ratio is over here, where 1, 2, 3, 4 offspring end up being RB or BR, so they all are 4 out of 4, or 4 to 4, or 1 to 1, or you could just say a 100% chance of getting all the same type of offspring. So what's the phenotype in this case? All of them would be purple in color because they appear to be blended. This is incomplete dominance. Like red and white makes pink. Like black and white could make gray. This is where we have a phenotype that is new and different than the parents' expressed phenotypes. So this Punnett square is just like a standard classical Mendelian genetics Punnett square, but we have capital letters to represent each trait, and a combination of both of them for the phenotype makes something new and different. Purple. All 4 offspring are purple in this case. Now we want to cross a purple with a purple. Remember that there is no purple allele if we were talking about red and blue making purple. So purple we're going to make as RB, and the other parent is also RB. If we do a Punnett square to represent the red and the blue, if we represent the other parents as red and blue to be purple, what are our offspring going to end up this time? What's the genotypic ratio? There's a 1 to 2 to 1. This is just like classical Mendelian genetics. What's the phenotypic ratio? This is not like classical Mendelian genetics. This time we end up with 1 being red, 2 that are purple, and 1 that is blue. That's because it matches the genotype of RR to RB to BB. This one has to be red in color. This one, there's actually 2 of them, here and here. These are your purples. These are going to be purple in color. And lastly, there's 1 that's BB, even though that kind of looks like an R, my fault. That is a BB for blue. So you end up with 1 red, 2 that are purple, and 1 that is blue. This is a new genotypic and phenotypic ratio for incomplete dominance. Now it's your turn to finish out your guided notes and to finish the questions in the Edpuzzle. Here's incomplete dominance. Tall and short makes medium. Here's another example of straight hair and curly hair. Straight and curly, if it's incompletely dominant, what would you get? What about if you cross 2 heterozygous parents for an incompletely dominant trait, like wavy cat fur? As a review from Mendelian classical genetics, cross a hybrid with a hybrid. For new incomplete dominance, what happens if you cross hybrid and hybrid? Don't forget to check out this vocabulary. Remember that they're on Quizlet. Thanks for being here. Thanks for watching.